Hi, everyone. We'll get started. On behalf of the UCLA Clinical and Translational Science Institute and the UCLA Cardiovascular theme, it is my pleasure to introduce today's lecturer, Dr. Barry London. Dr. London concomitantly attended Harvard College and Hebrew College, majoring in chemistry and physics and Hebrew literature. This was followed by MD PhD studies in the MSTP program at Albert Einstein College of Medicine. Then he completed medicine residency and cardiology fellowship training at MGH. Dr. London was subsequently recruited by the University of Pittsburgh, where he rose up the ranks to director of the Cardiovascular Institute and chief of the Division of Cardiology. In 2012, he was then recruited by the University of Iowa, Carver College of Medicine, as director of the Division of Cardiovascular Medicine and the Potter Lambert Chair of Medicine. He also serves as director of the Abboud Cardiovascular Research Center. His laboratory studies the molecular and genetic basis of sudden cardiac death in animal models, human families with inherited rhythm disorders, and populations at increased risk of sudden death. Additional areas of research include mechanisms of excitation-contraction coupling in cardiomyocytes and novel tools to image electrical activity and arrhythmias. Moreover, his group was the first to engineer mouse models of long QT syndrome. Dr. London has published over 110 original articles in over 50 reviews and editorials. He serves as guest editor for Circulation and the Circulation Electrophysiology and Genetics subjournals. He also completed an 8-year tenure as editor-in-chief of the Journal of the American Heart Association. Dr. London has been recognized by countless honors and awards and currently serves as president of the Association of University Cardiologists. Please join me in welcoming Dr. London, whose lecture is entitled Metabolic and Sex-Based Determinants of Arrhythmias and Sudden Death. Dr. London.
So thank you all for inviting me. When I left Iowa yesterday afternoon, it was 30 degrees with a 30-mile-an-hour wind and snow flurries in the air. This is better. So yet another reason I thank you all for inviting me. So I'm going to talk about metabolic and sex-based determinants of arrhythmias and sudden death. And I'm going to tell two different stories that I think converge, and I don't know exactly how they converge yet. This is the first time I will have given most of this talk. So, you know, it is not a completely formed, you know, finished story yet, unlike many of the talks that I've given. But I think it's intriguing and interesting, and we'll see where it goes. So I study cardiac arrhythmia as I have for the last 30 years. They remain a major cause of death, of illness. There are at least 250,000 sudden deaths a year in the United States. Our pharmacologic treatment for arrhythmias is not very advanced. We don't have drugs that really prevent sudden death well. We have devices that prevent sudden death very well, but they have all the problems with a $30,000 device that is implanted and affecting quality of life, etc. And we can't deploy them to all the people who need them. Now, most arrhythmias and sudden death happen in people with structural heart disease, people who have had or are having an MI, people with non-ischemic cardiomyopathies, viral myocarditis, subgroups, including people with radiation exposure, and I'm going to the heart, and that's one of the things I'm going to talk about today. Now, there are these rare inherited arrhythmia syndromes like long QT syndrome and Brugada syndrome, which my group has spent a lot of time studying. And they're important because we can learn a lot about how the heart works for them. And again, I'm going to talk quite a bit about Brugada syndrome today. So today is going to be Brugada and radiation, not, yeah, actually in that order. So this is a list of the inherited arrhythmia syndromes. They include long QT syndrome, short QT syndrome, Brugada, and some very rare versions of common syndromes like familial atrial fibrillation. AFib is common, familial, true genetically determined AFib is quite rare. These are the arrhythmopathies. We don't want to forget that there are also a lot of cardiomyopathies that come free of charge with lethal arrhythmias, like hypertrophic and dilated cardiomyopathy, ARVC, non-compaction, restriction, etc. Now, all of these are rare. The question is how rare. The most common of these conditions are hypertrophic and dilated cardiomyopathy with incidence in the 1 in 500s. Of the inherited arrhythmia syndromes, 1 in 2,000 is long QT, probably 1 in 2,500 to 1 in 5,000 is Brugada, and the rest of them are more rare than that. So these are rare syndromes. Why study rare inherited arrhythmia syndromes? It's two good reasons. One is, although they are rare, you can make a huge impact in the families that they affect by preventing family members from dying suddenly. And then the other reason, which I already touched, is we hope that we're going to learn something about mechanisms that's going to teach us something about the more common types of arrhythmias that happen when you have your MI or when you have heart failure. And that's already happened, and I think most of us bank that it will continue to happen over the next couple of years. I'm going to spend about 2 minutes reminding you about a little painful electrophysiology. Feel free to forget it once I am done reminding you of it. So heart cells are batteries, and they are, as are most of the cells in the body, and they live at minus 90 millivolts inside to outside. Why is that the case? That's because you have 140 millimolar of sodium in your blood and 20 millimolar intracellularly, 140 millimolar potassium inside cells, and 5 millimolar potassium in the blood. That's because you have a sodium potassium ATPase that pumps potassium into cells and sodium out of cells. You put channels in the membrane that are proteins that are permeable only to one ion, not both a cation and anion, but in this case, just a cation, potassium. Potassium 150 millimolar inside, 5 outside. Potassium flows down its concentration gradient, leaving behind a negative charge. There's something called the Nernst equation that tells you when you get to equilibrium. And if you plug in 140 and 5, you'll get minus 90 millivolts. So when the cell becomes minus 90 millivolts, there's no longer bulk flow of potassium out of cells, and you're at steady state with the inside minus 90. If you are a liver cell, the electrical life of a liver cell is you are at minus 90 millivolts until your host goes out drinking and you die and you go to 0 millivolts. That's the electrical life of a liver cell. If you are a nerve cell, a skeletal muscle cell, a heart cell, you use changes in voltage to signal and to do things in the heart to initiate every action potential and every heartbeat. How do you do that? You close the potassium channels, you open sodium channels. The sodium rushes in, positive charges because it's 140 outside, 20 inside. Voltage goes up to about plus 10 millivolts. You then close the sodium channels, you open some calcium channels. You sort of hang at steady state for 0.3 of a second. You then open a new set of potassium channels, and you go back down and reset the process to begin the next electrical activity of the heart. That is called an action potential in the heart. It happens once a second at a heart rate of 60. Now, action potentials spread from cells to cell, and the surface manifestation of that is the electrocardiogram. The P wave is the spread of electrical activation across the atrium. Halfway through the P wave, the upper half of the atrium of all the cells are at plus 10 millivolts. The bottom half is still at minus 90 millivolts because the excitation wave hasn't gotten there yet. You then have a pause through the AV node, which is a delay station. You then have the QRS complex, which is the spread of electrical activity across the ventricle, the isoelectric ST segment, when the whole ventricle is depolarized, and then the T wave, which represents asynchronous repolarization of the ventricle. If you have, so most of the arrhythmia syndromes are abnormal EKGs. Let's take long QT syndrome as an example. What causes long QT syndrome? The QT interval is too long. Why is the QT interval too long? Well, most likely reason is the action potentials are too long. Why are the action potentials too long? Well, either the potassium channels that terminate the action potential don't open properly, or the sodium channels that start the action potential don't close properly. And that is, in fact, the molecular basis of long QT syndrome. which Mark Keating identified, 25 years ago. It is, potassium channels that don't work or sodium channels that don't close. We're not going to talk about long QT syndrome today. This is just to note that the change in voltage and the opening of sodium channels and then calcium channels in the heart triggers the release of calcium from the internal stores. That calcium then binds to actin and myosin, which causes the cell to contract. The calcium gets pumped back, the channels close, and potassium channels open, and you reset for the next heartbeat. Okay, so today we're going to talk about, for the first half, about Brugada syndrome. So Brugada syndrome is one of these rare inherited arrhythmia syndromes, as I mentioned. It's characterized by a right bundle branch block pattern on the EKG, along with ST elevation in leads V1 to V3. It looks like you're having a heart attack of the anterior wall of your heart, complicated by a right bundle branch block, and that is in the differential when people show up. There is variable EKG expression. So somebody can have a completely diagnostic Brugada syndrome EKG today and a completely normal EKG tomorrow. Nobody fully understands that. Fever makes it worse. So people will often present with the EKG pattern and or arrhythmias, sudden death, when they have the flu or when they have a high fever, when they have COVID, which we've seen. Again, the rationale for that isn't completely understood. Brugada syndrome has minimal but some structural heart disease. If you look really, really closely at the septum, either by MRI or by biopsy, you will see a little inflammation, a little scarring, etc. So it's not correct to say there's no structural disease, but there's not much relative to the phenotype. Presents as fainting, sudden cardiac death, can present as sudden infant death syndrome, etc. And it's most common in Southeast Asia, parts of Thailand, Vietnam, China, where it's actually the most common cause of sudden death in young, healthy men, sometimes competing with trauma in the region. Now, some interesting practices have happened, but let me say that in the entire inherited arrhythmia field, there are no randomized controlled trials pretty much of therapies. There's been a couple that have been tried that haven't been done. In parts of Thailand, in families where young men are affected by sudden death, and usually at night when they're sleeping, one of the therapies that has come up is that young men would dress up as women when they went to sleep, trying to fool the gods that are causing sudden cardiac death in their families. No randomized controlled trials as to whether that works either. I'm guessing it doesn't, but I guess not completely sure. Anyways, so it is, but it is a bigger clinical problem. We don't have an explanation why it is so common in that region. It's not simple, oh, there's a gene mutation that happened in that region that affects all the people. It's something complex and probably genetic and or environmental. Brugada syndrome has an autosomal dominant inheritance pattern, variable penetrance, and male predominance. 80% of clinical cases are men. 80% of sudden death happens in men. There's some explanations related to other ion channels that have been purported to explain this, but I'm not sure that they're correct, and I will propose an option B today. Mutations in the main cardiac sodium channel are responsible for about 20% of cases. So this is an inward current or a sodium channel deficiency syndrome. So this is not enough depolarizing current to start the action potential. Now, unlike a lot of the other inherited arrhythmia syndromes, it's pretty clear that Brugada syndrome is polygenic, that with the exception of mutations in NAV1.5 that take out more than half of the inward sodium current, none of the other genes by themselves are enough to take out enough sodium current to give you the clinical syndrome. So you either need multiple genetic things or some genetic and environmental or pharmacologic things to bring out cases in most of them. There are common SNPs that have been identified that help precipitate Brugada syndrome. Families have been identified that have more than one mutation that have it. And as I mentioned, at the moment, only mutations in SCN5A, NAV155, the cardiac sodium channel, reach the threshold of causality, according to a recent sort of overview that was published by Dr. Husseini in Circulation a few years ago. I will suggest to you that there really probably should be 2 genes, not 1, that fall into this category. This is the list of genes that have been associated with Brugada syndrome. They all decrease inward current or increase outward current, which functionally has the same molecular mechanism. There's a number of other sodium channel related genes that sort of have been identified for which there's a little bit less evidence. And I mentioned that there's gene modifiers that bring out the condition. 2 mechanisms have been proposed for the arrhythmias in this condition. The first one was proposed by Ancelovich, and it's the more cool one, even if it's not the right one. So I'll go through it. So action potentials in the outer wall of the heart, the epicardium, are shorter than they are at the inner wall of the heart, the endocardium, because there's more of a polarizing outward current, early outward current during the action potential, the transient outward current in the epicardium. And that's what leads to the T wave on the EKG. If you don't have enough inward current, it has little effect on the epicardium, but on the endocardium. But on the epicardium, you can get an aborted action potential, which isn't full. So what ends up happening is the inner wall of the heart is already back at minus 90 millivolts, while the outer wall of the heart is minus 90 millivolts, while the inner wall of the heart, the endocardium, is still depolarized, and you can get spread of depolarization from the endocardium to the epicardium, and from spread of depolarization from the epicardium, from the endocardium to the epicardium, leading to what we call phase 2 reentry and arrhythmias. So, and that also explains the ST elevation, which is current flow from the endocardium to the epicardium. Now, a second version is just sodium channels are important in conduction in the heart. And if you have very slow conduction or failure of conduction in the septum, you would get the same functional phenotype with ST elevation and the potential for phase 2 reentry. And there's actually growing evidence that this may be the more common one, although these 2 mechanisms are not mutually exclusive. How do you treat Brugada syndrome? Well, you want to avoid things that block drugs that block sodium channels, because you already have that problem. The mainstay of treatment are defibrillators, and the question then is who warrants a defibrillator and who doesn't. If you've had aborted sudden death, it's easy, you get a defibrillator. If you're known to have Brugada syndrome and you either have ventricular tachycardia, ventricular fibrillation, or what sounds like arrhythmic syncope, easier said than done, then we usually put in a defibrillator. People with a baseline EKG that clearly shows a type 1 diagnostic pattern of Brugada syndrome are clearly at increased risk, but whether it's high enough to warrant a defibrillator is debated. And I note that defibrillators are really the only proven therapy for prevention of sudden death. You can do the opposite thing. If you don't have enough inward current, you can decrease the outward currents, which quinidine is, a transient outward current blocker, and does make the EKG better in some people and can stop arrhythmic storm. Similarly, electrophysiologists, I don't know whether we have any in the audience, can ablate anything. So certainly ablating the septum, which is the part of the heart that gets in trouble, is done, and again can prevent VT storm and make the EKG look better. But none of these have been proven as primary prevention for sudden death, and shouldn't be used as primary prevention for sudden death. So summarizing the clinical piece of this, Brugada syndrome is a disorder of depolarization of cardiac myocytes. There's less depolarizing current, and that leads to loss of the action potential plateau and or conduction failure in the septum and the RV epicardium, leads to transmural current flow, ST elevation, and reentrant arrhythmias. Questions that are unknown about Brugada syndrome. Why does the EKG pattern vary from day to day? Why do fevers exacerbate the pattern and arrhythmias? Why is the phenotype male predominant? And I'm going to suggest the following hypothesis, that this is evidence of a metabolic link of, channels to cardiac arrhythmias, and that this metabolic link is sex dependent. And that's what I'm going to try to give you some evidence to convince it is. This is how I got into it. So this is the EKG of a 55-year-old man who lived in Erie, Pennsylvania. He had no family history of anything, no personal history of anything. He was having lunch one day and he fainted into his egg salad sandwich. His colleague who was with him noted it. He woke up about 15 seconds, feeling fine. His colleague called an ambulance and had him go to the local hospital. They did this EKG. They said, you're having a heart attack on the front wall of your heart. He said, I feel fine. They said, you need a cath. He said, okay. The cath was fine. They did an echo. It was fine. They scratched their head a little bit and they called the head of electrophysiology in Pittsburgh, Kelly Anderson, and said, we have this. Do you think it's arrhythmogenic right ventricular cardiomyopathy? Could you guys do an MRI on him? And this is back in 1996 when the reports of Brugada syndrome, not called that at that point, were a single paper in an obscure journal with 7 cases of EKGs that looked like this. And Kelly said, this is that right bundle branch block ST elevation thing that was in the journal nobody's ever heard of. Send him down. And he asked one of the young electrophysiologists, Raoul Weiss, to see the patient clinically. And he said, well, we'll check his kids because in that original paper, a couple of them were related to each other at the time. So the guy came down to Pittsburgh and 2 of his kids came with him and they both had the same EKG that he had. And Raoul called me and said, Barry, you study arrhythmias. Do you want to study this family? And I studied mouse arrhythmias and had never done genetics on a human or anything similar to it. But I heard the story and saw the EKG and said, maybe I should start. So anyways, clinically, the question was, what do you do? So at the time, there was very little data. And Raoul offered the patient a defibrillator, and the patient, sort of said, okay. 6 months later, he wasn't feeling well, maybe had a virus, and his wife was driving him down to Pittsburgh for a checkup, and he fainted twice in the car. And that is his defibrillator telemetry before it shocked him, the 2 times out of ventricular fibrillation. This does actually show one of the problems with these inherited arrhythmia syndromes. That is 17 years ago. He has not had another event in 17 years. And we know that because he has a defibrillator and hasn't picked up anything in 17 years. So at age 55, he has a fainting spell. At 56, he has 2 syncopal events with something that might have killed him and nothing else for the next decade and a half. So these are rare events that are really hard to predict. And what caused this? What caused his clinical events? Was it the egg salad sandwich? Was it a virus when he wasn't feeling well? Really hard to tell. But so anyways, so I met with the proband when he came down and said, do you have any family? And he said, yeah, I have 10 brothers and sisters. And I said, well, that's good. Where do they live? And he said, well, 8 of them live on my street. And I said, well, that's even better. So he got a van, took an EKG machine up to Erie, and had 9 affected people the day, the first day we went there. Ultimately got more. This is the pedigree. Squares are male, circles are female, affected is shaded in black. You'll notice male predominant. You'll notice that there's an age dependence here, and that in this family is because the bundle branch block pattern develops with age. The J-point elevation, the ST elevations here in young people, but they don't have the bundle branch to get affected until they're older. We went ahead and did old-fashioned positional cloning and identified a locus on chromosome 3p, near but not the same as the cardiac sodium channel NAV15. about 10 million base pairs away or 20 million base pairs away from NAV1.5, had a large score that was consistent with linkage. We did mapping at the time, and we've since gone back and done SNP chip mapping to localize the area to a 3 million base pair segment on chromosome 3p24. We looked in, what genes are there. We did whole exome sequencing looking for, potential mutations and identified, a potential genetic cause. The only non-synonymous variant in the region of interest that linked with it was in an previously unknown gene called GPD1L, or glycerol 3-phosphate dehydrogenase 1-like gene. So a mouthful. It was a putative gene identified by the Human Genome Project with no known function. RNA is 4,000 base pairs long, encodes a 351 amino acid protein, 8 exons. It functions as a dimer. It is homologous to a known protein called GPD1 and somewhat less homologous to another one called GPD2 that I will talk about in a couple minutes. They all catalyze the reaction dihydrogenase phosphate to glycerol 3-phosphate. They are NADH requiring redox genes. GPD1 is known to be important in metabolism and an assortment of other functions. GPD1L, unlike GPD1, has a membrane binding domain, which is interesting. They both have NAD binding domains. The mutation is in a conserved amino acid. If you look in the big databases, it is rare, but not that rare. Its frequency is about 10 to the 4th, which is part of the reason why people question whether it is a causative gene for Brugada syndrome. It's completely conserved phylogenetically. We said, Brugada syndrome is a sodium channel problem. This gene probably decreases sodium currents. Let's check. So we took wild type GPD1L. Well, so these are artificial cell system hex cells. Yeah, we express sodium channels in them. They give you an inward current. We add wild type GPD1L. They give you the same inward current. We put in the mutant, you get less inward current. You can see that, so these things appear to decrease the amount of inward current by about 60% when you put in the mutant. Well, those are, human embryonic kidney cells. What does that mean? So we did the same thing in rat cardiac myocytes. You put in A2ADV GPD1L, you get a marked decrease in the amount of sodium current. So these decrease inward sodium current. Okay, what does it do? And what does GPD1 do? So this is something called the glycerol phosphate shuttle, something I guarantee no one in this room has ever heard of. But it is important. So you all know we have, the Krebs cycle and, sort of how we make energy. And when the Krebs cycle doesn't work in myocytes, you rely on glycolysis, and you need glycolysis anyway. And glycolysis, as you know, only makes 2 ATPs, and that's why it's not as good. But that's actually not completely true. So glycolysis not only makes 2 ATPs, but it also makes an NADH. It makes a reducing equivalent every time you go through the cycle. If you build up reducing equivalents in your cell will die. So you can't do that. And what the cell does is it takes the reducing equivalent, puts them into the mitochondria, and gets 3 ATPs out of the mitochondria for each time it puts a reducing equivalent in. So this improves the effectiveness of glycolysis, and it prevents the cells from dying. How does it do that? That's what GPD1 and GPD2 do. GPD1 takes these reducing equivalents and passes them to GPD2, which is a mitochondrial, it's encoded on the genome, but it's a mitochondrially located enzyme on the inner surface of the mitochondria that passes the electrons from the cytoplasm into the mitochondria. So that is what the glycerol phosphate shuttle does. And this is GPD1 and GPD2. Now, GPD1L is catalytically the same as GPD1, but it is located at the membrane usually. Okay, so next question. What I told you that GPD1L mutations decrease sodium currents. How do they do it? Well, we noted that there was no change in the kinetics of the currents at all. So we thought that this might be there are fewer channels getting to the membrane if you have the mutation in GPD1L. So to do that, we tagged the extracellular side of the channel with a flag tag so that we could pick it up really cleanly with antibodies. The channels still work well. We expressed them in in hex cells again, and you can see beautiful outlining of the membrane. And we then counted the luminescence of cells that got wild type versus mutant GPD1L. And you see there's fewer channels that make it to the membrane, in the mutant. So it looks like it's a trafficking problem. Well, where's GPD1L? I told you it had a membrane binding domain. Is it at the membrane? And so we did it two different ways. We pasted GFP, a fluorophore, onto green fluorescent protein, onto the C-terminus of GPD1L, and you can see it lights up the membrane. We also just immunostained the cells looking for GPD1L with an antibody we made. And again, GPD1L is present at the membrane. It's also present in the nucleus and to some degree in the cytoplasm. We did the same thing with the mutant A280V. And you can see it in the cytoplasm in the nucleus, but you can no longer see a membrane signature of it, in either way this was done. This just shows an overlap of GFP with the staining for GPD1L. And you can see in wild type cells, orange is the overlap. You can see staining of the plasma membrane. In the mutant, you cannot see it, and you see overlap in the nuclear membrane. Don't know whether it's doing anything there, but nothing in the plasma membrane, suggesting that GPD1L, the mutant, isn't functioning properly and isn't going where it's supposed to be. Well, we are a knockout mouse lab, so the next thing to do is to go ahead and make some mice. So we made a global GPD1L knockout mouse. Note to self, the homozygote is mostly embryonic lethal. So although we did get one homozygote, in multiple years, for the most part, all we have to study are the wild types and the heterozygotes. If we look at total GPD1L protein, the heterozygotes have half as much. The mutant has none. Sodium channel NaV1.5. This actually looks like there's a little upregulation of NaV1.5, sort of globally. If we do a membrane PEP and only look at proteins in the membrane, here again, you see GPD1L still shows up. So GPD1L is present at the membrane. There now appears to be a little downregulation, but not that much of NaV1.5 at the membrane, except in the one homozygote where you can barely detect anything, and probably part of the reason that these mice don't do well. And if you patch myocytes from the heterozygote mice, you get about half as much current. So in the mice, having half as much GPD1L gives you about half as much current. So that's consistent with something that could cause Brugada syndrome. Heterozygote mice aren't that much fun to study, so we went ahead and made cardiac-specific GPD1L knockout mice. And this is where we started to see something that we completely did not expect. So at 9 months of age, male mice developed a dilated cardiomyopathy. And you can see end diastolic volume goes up and systolic volume goes up, ejection fraction goes way down. Now, turned out that when we first presented this, we had 4 pups and they were all male, and this is what we presented. But we went back and said, we need to fill out the cohort and let's get some females in here, etc. This is at 9 months of age. At 6 months of age, the males start to have a phenotype. At 9 months, they have a robust heart failure rate. Oh, yeah, they're dead. Females at 9 months, absolutely nothing. Hearts are normal sized, absolutely normal ejection fractions. So this is sex specific. Now, we've carried a small number of females out longer, and they start to develop something about 1 year and a third. At about 15 months out, they start to develop a myopathy, but way delayed. compared to the males. Well, how about electrophysiologically? Do they have Brugada syndrome? Well, Brugada syndrome is hard to replicate in a mouse, but they do have a wide QRS and some PR prolongation to go along with the clinical phenotype. They have spontaneous ventricular tachycardia in the homozygote mice, but again, in the males. If you look, the males have some PR prolongation, marked QRS prolongation. Females, absolutely nothing. So this is a sex-dependent phenotype. Note to self, in a condition that happens in men and not in women, for the most part, at least 5 times more in men than in women. Okay, are there any clues that we can get as to sort of how GPD1L is actually causing changes in trafficking to the cells? Well, our group published about 5 years ago a paper showing that sodium channels are acetylated in the domain 3,4 intracellular linker, and that deacetylation promotes trafficking of sodium channels to the cell membrane. That deacetylation is done by sirtuins, in this case SIRT1, which are deacetylase, NAD burning deacetylase proteins. So we said, is this involved? So we checked whether SIRT1 and GPD1L co-immunoprecipitate, and they robustly do. We checked SIRT1 deacetylase activity in cells that were given wild type versus mutant GPD1L. And there is actually less deacetylase activity in cells given the mutant GPD1L. We then went to the mice and said, are the sodium channels more acetylated? And by Western blot, you can see sodium channels are more acetylated. And by immunohistochemistry, you can see that sodium channels are more acetylated. So one of the mechanisms here is this. Now, the next step you ask me is, how is this happening? And I don't have a particularly good answer for that. Now, we also, though I'm not going to show today, have some hints that there are some other post-translational modifications of the cardiac sodium channel that are affected and not happening normally in the presence of mutant GPD1L, specifically simulation. But more to follow on that. So this is what I think is going on, at least. I think that you have the glycerol phosphate shuttle here. And I think GPD1L is a membrane version of this, whose purpose in life is to take the metabolic temperature of a cell, of what's going on, and feed that into how ion channels make it trafficked out to the membrane. If you have a mutant form of GPD1L, it can affect sodium channel post-translational modification and can affect whether sodium channels get to the membrane. So mutations of GPD1L can directly affect, sort of how channels work and presumably cause inherited arrhythmia syndromes. But they can also do it indirectly. A mutant GPD1L could go back and muck up the glycerol phosphate shuttle, change NADH, NAD ratios in cells, although we've looked and we don't see that in our mice. So I'm not sure that is happening, but it could still change cellular redox state, cellular energy metabolism, mitochondrial metabolism. And I think that this probably underlies sort of some of the fever effects that we see. So to summarize the first half of the talk, GPD1L mutations cause Brugada syndrome in a large multi-generation family by decreasing membrane sodium channels and the sodium current. I suggest that there's more than enough evidence to believe GPD1L is a causal gene for Brugada syndrome. GPD1L mutations may impact channels either locally at the plasma membrane or through changes in cellular energy or redox metabolism. And I see no reason that this couldn't explain how or be part of the story as to why there's a male predominance for Brugada syndrome. And we need to still figure that out. Okay, I'll hurry through the last part of this. So this is, we got into studying radiation in the heart when I was in Pittsburgh. So therapeutic radiation happens. People get radiation for cancer. People get intentional exposure. Some of our electrophysiologists, I believe at your site too, are looking into ablations using radiation. And there's the potential for environmental exposure. And although the heart isn't the main problem, it could be one of the problems. Radiation exposure to the heart causes a litany of problems to heart failure, pericardial disease, valve disease, coronary disease, and arrhythmias and sudden death. Mechanisms include reactive oxygen and nitrogen species, apoptosis, senescence, endothelial dysfunction, inflammation, fibrosis, and changes in autonomic nervous system function. So a lot can go on. A collaborator in Pittsburgh identified that NOS1 mice were more sensitive to radiation than the NOS1 knockout mice than wild type mice. And he noted by video recording that they seized and died all of a sudden. He came to me and said, "Is this cardiac?" And I said, "I don't know, but we can check." So we went ahead and radiated mice with telemetry, with implantable telemetry monitors, so we could see what was happening to their rhythms. Yeah, I won't talk about it, but we did it with and without a radioprotective agent that they were testing in Pittsburgh at the time. Followed them, gave them a lethal dose of radiation, 9.5 gray, followed them for 9 days. You can see NOS1 knockout mice die sooner than wild type mice. Interestingly, wild type mice, they get sick, their heart rates fall, they die, non-cardiac causes. NOS1 knockout mice, their heart rates start a little bit lower, don't change at all. They go into heart block, they seize and they die. So this actually is cardiac death. They have, some nice arrhythmias if you want to look at arrhythmias. This is just sort of the same, sort of following the telemetry and the EKGs, between day 0 and day 9. You can see wild type mice develop PR prolongation and QRS prolongation. The NOS heterozygote mice, less so, but still a little bit of PR and QRS prolongation, but no QRS prolongation. And NOS knockout mice, sort of the same thing. So less, some PR prolongation, but no QRS prolongation. So electrophysiology is different, and NOS1 appears to be playing a role in the nodes that we didn't know it used to be playing. So we went ahead and said, these are short-term experiments. They're not really relevant to radiation effects in humans, where you irradiate it and you see a phenotype a year later or 10 years later. So we developed a system where we give cardiac-specific radiation, 20 gray, to the heart contoured only to be in the heart by CT, a little bit of lung exposure nearby, but not that much. We again tested the radioprotective agent, which I'll just say didn't work, and we'll leave it at that. And this is the results of the experiment. And again, not something that we expected. Heart weight, body weight, so hypertrophy. So wild type and heterozygote NOS knockouts. We can't do the homozygotes. They don't live long enough to follow them for a year, even without radiation. So male mice, you give them radiation, they get hypertrophy. Heterozygote mice, they're hypertrophy to start with, and they don't get any more hypertrophy. Female mice, absolutely nothing. You give them radiation, absolutely no effect in heart weight body weight ratio. Again, the heterozygotes are a little, have a little hypertrophy compared to the wild types. If you look at lung weight body weight, a ratio of congestion, here, male mice, wild type and heterozygote mice get lung congestion. Female mice get just as much lung congestion. This is fibrosis. This is diastolic dysfunction. So you're giving female mice radiation. They don't hypertrophy, but they do get lung trouble. What else do they get? The males get dilated cardiomyopathy. The females don't. So males and diastolic volume goes up, nothing in females. Ejection fraction goes down, nothing in females. This is sex-specific, effects of radiation, of high dose cardiac radiation in mice. Nothing that would have suspected. Well, what if you do this? What happens in the heterozygote NOS1 knockouts? Well, this is different. So here, both the males and the females, the heart gets smaller. It doesn't dilate. It actually gets smaller, and ejection fraction fall in both sexes. So eliminating half of NOS1 eliminates the sex-dependent phenotype in this, suggesting that NOS1 is part of that phenotype. We looked at EKGs, and there wasn't actually all that much here, except that in the heterozygote NOS1s, in the heterozygous NOS1s, PR interval actually falls in both males and females. So again, NOS1 is playing a role in the node, and radiation is interacting with what it does in the node. I can't say I know exactly what it is just yet. This is something that was remarkably striking. So we went ahead and looked and started asking the question, well, what's happening to energy metabolism? So this is mitochondrial complex 1 and complex 2 activities, which our collaborators do very well. And so if you look at males, the radiated group, there's no change in complex 1 activity, and there's an increase in complex 2 activity. That usually means that there's a downstream blockage and it's backing up behind it. If you look at females, there's a huge increase in complex 1 activity, and complex 2 activity is gone or almost gone. So this is, the backup behind it. We went ahead and looked at one of the proteins in complex 2 to see if it was doing what we expect. And you can see in males, there's an increase in complex 2. And in females, there's a slight decrease, not enough to explain the complete loss of activity, but it's presumably one of the other proteins that are part of that complex. And I told you heterozygote, NOS1 heterozygotes don't have it, and they don't have the energy signature either. There's really no sex-dependent difference between the two. Well, so NOS is involved. What does NOS do in the heart? And so this is the first attempt to look at that. So this is staining of 3-nitrotyrosine, which is a, so one of the modifications from one of the nitrosylation products of the presence of nitric oxide. At baseline, you can see purple is the 3-nitrotyrosine. You can see 3-nitrotyrosine pretty uniformly. This is the right ventricular free wall. This is the septum. You can see that it looks like there's less in the middle of the septum than there is at the edge of the septum and in the RV free wall. And this is quantification of that. If you give radiation, now the center of the RV and the center of the septum are devoid of 3-nitrotyrosine. And you can see the difference here. So radiation is affecting nitric oxide and nitric oxide byproducts through the heart. And this is likely part of the story, but we don't know the answer yet. So to summarize, NOS1 expression modulates the effect of ionizing radiation on the cardiac conduction system in mice. Radiation effects on the electrical and mechanical functions of the heart are sex-dependent, not something we expected. Decreased levels of NOS1 expression eliminate the sex-dependent differences, and they're actually deleterious to female mice. People think that reactive nitrogen species are bad for you. This suggests that they're actually protective, and they may be balancing lost reactive oxygen species, which are bad for you, and we're in the process of looking at that. And they suggest that changes in energetics are associated with functional changes, and that this may be causal, and this may be relevant to humans. Have humans been studied regarding sex-dependent differences in radiation? The answer is not well. Why? The prior studies that are out there are in Hodgkin disease. Most people with Hodgkin's who got mantle radiation 30 years ago were men or were boys, just because it's more common in teenagers and young men. You have to survive to be able to be studied. The survival, the setting where people get radiation and survive is breast cancer, and almost all breast cancer is women. So you can't do sex-dependent studies there. Only now are people surviving long enough with esophageal cancer and lung cancer who get radiation that affects the heart to be able to be studied. So there will be an answer as to whether this is relevant in humans, but it's probably a couple of years away and requires people to start going through the databases and see whether with modern therapy where you have several years survival in a reasonable number of people, whether this makes a difference. Future directions, clarify the roles of ROS and reactive nitrogen species, test radioprotective agents and how they work, and target them based on whatever mechanisms we find. Extending the studies to larger mammals and to humans exposed to thoracic radiation, and determining the relevance of sex differences sounds like a really good thing to do. And with that, I will stop. These are the people who did the work in my lab at Iowa and in both my lab when I used to be in Pittsburgh and Joel Greenberger's group, who've been really good collaborators. Thank you all very much.
Thank you so much, Dr. London.
That was fascinating, Barry, especially the Brugada part of the talk. I found really intriguing. I mean, you presented a very persuasive argument, but I still wasn't sold on how the reducing equivalence and maybe per se the metabolic activity is linked with the trafficking of the channels, or whether the protein GPD1 has an independent effect on, sirtuin activity as you showed. Because there are other ways the cells can deal with, for example, if you have the Krebs cycle down, you turn lactate to pyruvate, and that's how you get your NAD back. And so whether you looked into those things, and especially you did for the second half of the talk, but to sort of nail the argument, whether you actually looked at cellular respiration in the heart to see whether, the energy efficiency is down.
Yeah. Oh, sorry. That's right. I do have this. Yeah. So you're correct that there could be other mechanisms that, and that we haven't nailed down the mechanism, sort of by, that links, cleanly. This is the mutation. This is how it happens. Now, GPD1, and remember I said that Brugada syndrome is one of the more common of these. GPD1L isn't that. This is very rare. There are now 3 families with GPD1L mutations in Brugada syndrome that are out, and part of the reason there's been reluctance to accept GPD1L as a Brugada gene is because there just aren't that many of them out there. And, I mean, I would suggest that there really just aren't that many of them out there, and these rare inherited things, some of them are really ridiculously rare, and this falls into that category. This family, part of the explanation as to why they show up with Brugada syndrome is, I mentioned that there's these modifier genes that are common SNPs, and this family is unlucky in the sense that it carries a couple of the modifier SNPs that are linked to, because they're in the region around NAV1.5, and that's not that far away from GPD1L, they're actually partially linked to it. So they greatly overexpress sort of modifiers that would help decrease sodium current specifically in this family. But you're correct. There's a lot of other stuff that could be going on. Part of what we want to do is, we obviously have the mouse model. It's hard to do these experiments, and obviously in people don't want to give us part of their hearts and stuff. The mice aren't the most viable thing on the planet. They don't breed all that incredibly well. I think that, whatever we're doing to muck up, sort of their hearts doesn't make it particularly easy to breed. They have small litters, etc. So it's a bit of a pain to study, which has gotten in our way. But, but obviously there's something here, and I think that it is answerable, and you're right, I gave you a bunch of conjecture, and a little hand waving.
In your mutant mice, you clearly see sex differences. In male, you see the phenotype at 9 months, and in female, in 15 months. I just wanted to know, have you ever overexamined your mice to see if this protection is gone, or you treated your main mice with estrogen to see if we can.
A very good question. So the short answer is not yet. We plan those experiments both in the radiation. The next step is going to be to repeat the radiation experiments, be gonadectomized and repleted either with their native hormone or with the opposite sex hormone. So we haven't done that yet. We do know in humans, there's pretty good data that, that male is bad and female is good and it's hormonal. And we know that because there have been people who've gone through sex change operations and the EKG phenotype has tracked with the sex to which they changed. So it is likely hormonal, hormonally related.
Good to see you. Great talk. I'm interested in the early slides you were talking about the mechanism of the arrhythmia induction. Now, you mentioned phase 2 reentry, and then you kind of suggested that's probably not right. And then you talked about another one, which is just slowed conduction.
Yeah.
You referred to a paper, but I'm a little vague on how just slowed conduction
So
leads to that kind of
okay.
arrhythmia. And
Okay.
by the way, did you see a QRS prolongation?
So there is QRS prolongation. So I'm sorry if I wasn't clear. So phase 2 reentry is the mechanism, both whether it is from aborted action potentials or whether it is from failure to conduction. So if you think about it, the epicardium of the right ventricle and the septum, is not appropriately depolarized. It is repolarized. It's either repolarized because the action potential was ridiculously short and the action potential aborted, or it's repolarized because there was conduction block and the action potential never got there. And then you have current flow and phase 2 reentry with both of the two mechanisms. There is, we can do whole body EKGs on people. And, Yoram Rudy's group in Cleveland developed those. And of course, they quickly said, we can answer a question in Brugada syndrome on this. And they did it. And their data suggests that it is conduction failure to that area. Although, again, it's not all situations. And the two mechanisms could be hand in hand. Anselivich's group has great data in dogs and other animals that the sort of aborted action potential mechanism could be the mechanism that's involved.
Question for the NOS1 knockout, was that organ specific or is that whole body knockout?
No, so that is a whole body knockout. There is a cardiac specific NOS1 knockout, which we tried to get, and at least in the initial iteration failed around the COVID era to be able to get it. And we haven't gone back and tried to get cardiac specific NOS1 knockouts. And you're correct, it could be different. And obviously nitric oxide is produced by myocytes, but it's also produced by other cells that are in the heart. And how that is, I point out, although it's not in this half of the talk, that the GPD1L knockout behaves quite differently between the cardiac specific knockout and the global knockout, in the cardiac phenotype. And it does suggest that there might be, sort of interactions of other cell types, sort of in the heart that are, coupled electrically, et cetera, to the myocytes. So, so it's more complicated than just a bunch of myocytes.
Right, for the nitric oxide synthase, like it produces nitric oxide, which is a natural vasodilator. Are you also looking into the role, like how vasodilation might be playing? It's
a very good question. We just put a grant in on this, and yes, part of the grant was to, sort of look, to look at the vasculature too. And one of our collaborators is a vascular biologist who, sort of obviously has interest in this. There's no question that when you throw 20 grays of radiation at the heart, that you affect endothelial dysfunction, you cause endothelial dysfunction, you cause an assortment of vascular problems, whether you're human or whether you're mouse. And so the answer is sure. Could some of the sort of effects that I showed you in 3-nitrotyrosine be vascular? Of course they could. I mean, they seem to be the parts of the heart that are metabolically most active, that are generating the most tension, that have the most compromised blood flow by contraction. Those seem to be the ones that are most affected by radiation. So yes, short version.
Thank you very much, Dr. London. Thank you.
Thank you.